day, grade 12 learners, and welcome to Life Sciences Evolution Revision lesson. My name is Diniko Vuma, and this lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. Learners, today we are going to go head on with revision of those chapters that had given us teachings. So we'll be looking at genetics, we are going to look at cloning, and we'll also look, the bulk of our revision is going to be evolution. I hope you are going to enjoy this because the exam is just around the corner. So let us look at one example of our genetics problem. Genetics, remember, is complex. There's so much that you need to know. And just remember that genetics is carrying a lot of marks also for your life sciences paper too. You are writing 2 papers in prelim and at the end of the year. And there are a lot of marks in genetics for your paper too. We are just going to look at one example which had seemed to be a challenge to most of the learners. And we are going to look at the sex-linked genetic conditions. Here is an example that we're going to be looking at, which speaks about hemophilia. Hemophilia is not Greek to you. I hope you just remember that. If somebody is suffering from hemophilia, they do not have the blood clotting factor and that they experience internal bleeding. And remember, hemophilia can affect both males and females. Let us read together. Hemophilia is a genetic disorder that results in abnormal clotting of blood. It is caused by a recessive allele carried on the X chromosome. I hope you remember that the Y chromosome does not carry anything. And one reason is that it is small, it cannot carry any of the conditions. So hemophilia is recessive. That is why we're underlining recessive, because there is either dominant or a recessive condition. This one is recessive. The allele for normal clothing is X, capital letter H, and the allele for hemophilia is X and small letter H. You can see that the capital letter is representing the dominant part and the small letter represents the recessive part. The inheritance of hemophilia in a family is shown in the diagram. So we have got a diagram here that is showing us the inheritance of hemophilia. If I may ask you, what do we call this diagram, by the way, grade 12s? This is a pedigree diagram. So just remember that this is our pedigree diagram. In the diagram above, now we are looking at the questions. How many generations are shown? Before you even start answering the genetic problem or any problem that is given with a pedigree diagram, you need to familiarize yourself with what you see. On the pedigree diagram, we have the diagram itself, but important enough, we have the key. Look at the key, do not ignore it, because the key is going to give you clear guidance of how to engage with answering the questions that are asked. So if you check our key, there is saying we've got non-hemophilic female, it is the unshaded circle, and hemophilic female is the shaded circle. Then we've got squares, that's a non-hemophilic male, and the shaded one is hemophilic male. Then back to the question, how many generations are shown? How do we see generations? You see by the levels that are shown. So in other words, if you check here, this is 1 generation. And now we have got the generation of the offsprings. We have got the generation of the offsprings, right? And this is now the 2nd generation. Therefore, our answer to the question that is asked should be, we have got 2. you are able to identify, we have shown each other how to identify the generations. So this is the first generation, and we have got the second generation. 2 generations are shown. Then we carry on to how many females are carriers of hemophilia or heterozygous. By the way, now you go back to the pedigree diagram. You see this one is very simple. It has only 2 females. How do you see the females? By the circles. And now the question is, how many are heterozygous? They are carriers. So if we say that a female is a carrier, it means that this female should be having, this is not the answer because the answer is asking about the number. But for a female that's a carrier for the condition, they should be having this. That is a carrier. You can see they are not suffering from the condition because the dominant allele is masking the recessive one, but they are carrying the allele for hemophilia. So how many females? If you look at this female here, this female here has got 3 children with the male. And the 3 children are, if you look at this B, B is shaded. It's a male that is shaded, meaning that this male is carrying that is our male. It's the shaded male is suffering from hemophilia. But when you look at this one here, it's unshaded, meaning that this woman here should have heterozygous combination, this combination. That is why we've got B, and this small one here comes here for the male that is suffering from the condition. And if you check this other male here, the middle one, should be having X and capital letter and a Y. But because this male here is shaded, it means that this male here on our pedigree diagram is X and a small h and a Y. Therefore, this female here is going to be getting, because this female is not suffering from the condition. So this female here will be, because she's not shaded. So this female here is this, getting the capital letter from mommy and then getting the small letter from daddy. Therefore, our answer, you can now tell how many females are carriers. And our answer is 2. This female here is a carrier and this female is also a carrier. I hope that you understand the dynamics. So if you work on genetics problems, what you need to look at, especially in a pedigree diagram, look at the offsprings. Look at the outcome of the offspring. So that is going to give you a guidance of the genotypes of the parents or the generation before. The next question is, provide the possible genotypes. Provide the possible genotypes for individual A and B. So we are looking at, this is our individual A and we are looking at individual B. The good thing is that when you answer the previous question, we have engaged on looking at the genotypes in order to find out the females that were carriers for hemophilia. So if you look at this, you are supposed to do what? To provide possible genotypes. I'm advising you to check and remember the terminology in genetics, because if you don't have a good background of the terminology, you will not even understand. You can lose marks by maybe giving the phenotypes, and the question is specifically asking you to give the genotype. So as we have worked it out, let's see, this is B and B is shaded. And you know B that is shaded must have only one X and a Y, because that's a square that is shaded and suffering from the condition, meaning that this one will have a small h, and A, it's a female. So before you even think twice, you write XX to show us that it's a female, but this female is not shaded. However, this female had given this child a dominant allele. So this female here is, there we go, heterozygous. But then our answer is not heterozygous or homozygous. Let us look at the possible genotype or genotypes of individuals A and B. Yes, we got it right. So A, as per indication here, it's heterozygous, but now we wanted the actual genotypes, and B is a male, so you can see that B is carrying the small h. Beautiful. Let us see if we can just go further and work on a genetic problem based on hemophilia. The question says a mother A is expecting a baby girl. So you're coming back and looking at mother A. This is what you do, is expecting a baby girl. Mark this as well. Using a genetic cross, show the percentage chance that her daughter will have hemophilia. Look at the trick. They are asking about her daughter. And what are you supposed to give us? To give us the percentage chance that her daughter will have hemophilia. How many marks do we have in genetic cross? Basically, it's 6 marks. From these 6 marks, you will find that they are compulsory marks. The compulsory marks are the marks that will directly answer the question, and the other marks are the ones that are showing us the steps that you take when you do a genetic cross. So everyone remembers that it's a key. Remember, you have been taught about let me call it a skeleton. You have got an outlay or a skeleton of a genetic cross. Now you answer in context of what is asked. And in context of what is asked here, you answer in context of mother A that is expecting a baby girl, and you are looking for the percentage chance that her daughter will be hemophilic. For genetic cross, remember we must indicate the generation. So we've got our P1 generation. But when you put your P1 generation, just remember that we will have our F1 generation. It's a parental generation, P1, and filial generation, F1. You get 1 mark for indicating the two. If you put P1, for example, and you forget to put F1, you lose that 3 mark. Learners, each and every mark counts. You cannot afford to lose that mark. So let us check quickly. We have got the phenotype. We start with the phenotype. I want you to check. So it is hemophilic male. Okay, we have got a hemophilic male. That's a hemophilic male. And this male is hemophilic, you see. And now this female is non-hemophilic. As much as this female here is a carrier, so she's non-hemophilic. You are not allowed or supposed to say male suffering from the condition and female not suffering from the condition. You specify. Like in this case here, we're speaking about hemophilia. So you tell us that it's a hemophilic male that is crossed with a non-hemophilic female. You then immediately have your Mahala mark, free mark. That's your mark there. Then you go to the genotype. The genotype, you now look at the genetic makeup of the father. This male here is shaded, and therefore this is the genotype. It's X, small h, and a Y. Y, just remember, it's not carrying anything. Sometimes learners have got a tendency of adding something on the Y. Just remember every time that when you see XY and it's a genetic problem, Y is not carrying anything. Then we've got the non-hemophilic female, but this female we already know by checking the pedigree diagram that this female, because she's non-hemophilic, yes, we agree, but she's heterozygous because of the outcome of her children here in this generation. So this female here is going to be X, capital H, and X, small h. You score your mark immediately as well. Then that is your parental generation. Then you tell us that this is now meiosis. Meiosis is supposed to take place, and when meiosis takes place, we are going to now have what? Gametes, because gametes result in the meiotic division. So we are going to have gametes, and please be careful, extra careful. You can see here that the gametes that are indicated, there's a comma in between. So you must indicate clearly. You see there's X small h comma and a Y. There's also a small gap in between. These are technicalities of genetic crosses. These are the things that you need to keep an eye on, because once you forget, for example, to put a comma, or you forget to create that space, it's no longer gametes, and then you lose your mark for gametes. So we've got X small h comma and a Y, and then X capital H comma and X small h. And then there we go. You can also, if you want to, because some practices are different from what we have here, if you want, you can say that your gamete, for example, for the male, is this, and then you circle it, and then you come here and you write your Y, and then comma, then you come to the females. That's still fine. This is a genetic cross, which is for most learners a bit confusing, but for some they are used to it. The lines must be crossing correctly in order to get the marks for the offsprings. So you can see that the small x, the x and the small h here is crossed firstly with this part here of the female, the first gamete, which is X and capital H. And this is the outcome. You see the genotype, that's the genotype. Then you carry on, you go to the same one, and you come to the next one. And you have got X small h and X small h. This you do not just guess. You check that you have crossed, for example, you check that you have crossed, it's X small h, this is small X small h from the male, and then this was crossed with small X and small h from the female. That is why we have got this genotype here. If you just write genotypes anyway without checking how you have crossed the lines, you are losing your marks immediately. And the process continues like that. There we go, you cross that and you cross that, and these are the genotypes. Now, the phenotype that we have at the end is that we have got non-hemophilic female, we've got hemophilic female, we've got non-hemophilic male, and hemophilic male. Let's now go back to the genotypes and see if we understand what we mean from the phenotype. You check the genotype here, this one here. This is non-hemophilic, so you can relate. And you check this one here, X small h and X small h, that is a hemophilic female. Why do we see that it's a female? Because it's two X's. And we have got this male here with a small capital H and a Y. This male here is a non-hemophilic, because hemophilia is caused by a recessive allele. And then you come here, this is X and small h and a Y. It's a male, and this male is a hemophilic. But don't forget, we were looking at what? We were looking at the percentage chance that her daughter, we're talking only of the girls, her daughter will have hemophilia. Now the chance here is 50% chance, 50% chance. Somebody might be asking, ma'am, I don't understand, because in genetics, we are told that there are 4 offsprings that we get. So each one represents 25, 25, 25, 25%. But in terms of the specification of this question for the girls, it is 50%, because now, how many girls do we have from the first filial generation? We've got 2 girls. So if we are to get a percentage from the 2 girls, one girl will be 50%, the other girl will be 50%, giving us 100%. So when we have these 2 girls, we check. One girl is non-hemophilic and the other girl is hemophilic. Therefore, our answer is 50%. You can also see that there is an asterisk here, which is showing us this is a compulsory mark. Do not forget to answer what is questioned, because if you got everything, the ticks that are here are more than 6, but you will only get 5 marks if you did not include the compulsory mark. And just as I indicated to you, for you to have indicated parental generation and first filial generation. Again, you put them at the right place, you have your 1 mark. And for you to have said there's meiosis that is taking place, and when gametes are formed, now there is fertilization. Again, these two must be placed at the right place, or placed at the right place on the genetic cross. Then you have your other mark. We have got our 6 marks. This was well explained, I suppose. This is just the other way of answering the same question when Pinette square is used. So you can see that I'm not going to go through all this because we have looked at that. Meiosis, fertilization, we have looked at that. But now instead of the diagrams where you draw lines and cross, here we've got the Pinette square. And on the Pinette square, you can see the gametes. These are the females and these are the males. And the outcomes are here as you cross them. I personally find it a bit easier to use a Pinette square and not use the diagrams. However, all the learners have got the luxury to choose what works best for them and what they have been taught in class. Let us now move to something else, and this is about cloning. A farmer decided to have his best meat-producing bull clone. Best meat-producing bull cloned. The following steps were in the process. So you look at the steps that were done. In cloning, remember in biotechnology, genetic engineering, there are steps that are being taken in terms of achieving what the experimenter was looking for. Here we are looking at cloning. So the process has got different steps. So let's look at the steps. We've got a muscle cell that was taken from the bull and the nucleus was removed. Muscle cell is a somatic cell. So this is a muscle cell. It is a somatic cell. Think about other things as you go through this. Somatic cells, are they haploid or diploid? Body cells, somatic cells, they always have the double number of chromosomes. Therefore, somatic cells are diploid, right? You are thinking as you engage with the question paper. And the nucleus was removed. An ovum, now check this, an ovum was taken from a cow and the nucleus was removed and discarded. So here, you can see that we've got 2 nuclei. One nucleus was from the muscle cell that was removed, and then from the ovum, we've got the nucleus that was removed and discarded, thrown away. The nucleus from the muscle cell was placed in the empty ovum. The nucleus, and I want you to think, because we said the muscle cell is diploid, it means that this nucleus already has got diploid number of chromosomes. It has got diploid, its complete set. So the nucleus was removed from the muscle cell and was placed in an empty ovum. The ovum was given an electric shock to stimulate normal cell division to produce an embryo. The embryo was placed in the uterus of a surrogate cow, where it developed into a clone. So in other words, you see that we've got 3 individuals that were involved in this whole process. 1, the bull. What was taken from the bull? It's a muscle cell, which is a somatic or body cell with double the number of chromosomes. The nucleus has got double the number of chromosomes. The nucleus is removed and kept. Then we've got the cow, in which the ovum is removed, but the nucleus is discarded, thrown away. And because now the ovum is a gamete, it means that it has got haploid number of chromosomes, half the number of chromosomes. So now the third one that is involved is the surrogate cow. This is the cow that is going to carry the embryo. How did the embryo develop? Through the electric shock to stimulate cell division. I want us to start thinking that embryo that is developing here, do you think it will have a haploid or diploid number of chromosomes? Just think, this embryo develops from the ovum that was given a nucleus which was diploid. meaning that now the embryo has got complete set of chromosomes. It's growing. Now it's placed on the surrogate mother or surrogate cow, where it's going to develop into the clone. The first question is, what is cloning? What is cloning? And everyone remembers terminology is key. Cloning is the production of genetically identical organisms. So if I am cloned, it means that what is going to be the outcome? It's me exactly genetically. And the question is, what will be the difference between me, Mrs. Vuma, and my clone? The difference will just be age, because the clone would have developed into a mini me, but that's going to be genetically exactly the same as me. Then 1.2, explain why the nucleus of a muscle cell was used and not the nucleus of the sperm cell. Some of these questions, when we come across already, we have pondered, we have engaged with the stem of the question, and it becomes easy for us to answer. Why was the nucleus of a muscle cell used? Already part of it we discussed before we reached this question. And the reason is that a muscle cell contains all genetic material of the bull, complete set, and is diploid. It has got diploid number, so it is diploid. It has got everything, like full set. Whereas a sperm cell has only half of the genetic material, so it is haploid. You know, sperm cells and ova, they are haploid. They've got half the number of chromosomes. And the reason why the nucleus of a muscle cell was used, because it is carrying a complete set of the chromosomes that are needed for an organism to develop. 1.3, explain why the nucleus of the ovum was removed. I hope you remember the stem that it was removed, and what happened to the nucleus of the ovum? It was discarded. So it was removed and thrown away. Why was it removed? To remove the genetic material of the cow so that only the genetic material of the bull is present. And by the way, this farmer, what was the main reason of this process that is taking place here of cloning? The main reason is that this farmer realized that this bull is the best meat producer. So cloning this simply means that it is for market reasons. That if I can clone this bull, it means that I will have more and more exactly the same that can give me more meat production. So remember, the nucleus is the genetic material. So if the nucleus of the cow was removed and thrown away, the genetic material of the cow is removed, because we don't need anything. We have nothing to do with the cow that was used here, but we have all to do with the bull, which is best meat producer. Then state one benefit of cloning for one mark. This is a walk in a park. You have learned about the different benefits of cloning, of which you can recite them, but the examiner is only asking you for one benefit. So there are different benefits. One of the benefits will be to produce organisms with desired traits. For example, on this scenario, we are now getting organisms with best meat producing genes. So to produce organisms with desired traits and to conserve threatened species. So let's say that For example, this best meat producing bull was becoming threatened. You know, when organisms are threatened, they are nearing extinction. So it means that the whole species will be wiped out and will not have such anymore. And just remember as well that once we have organisms that have gone extinct, our biodiversity becomes minimized. We want to maintain the full biodiversity. And also to create tissues or organs for transplant. Cloning makes it easy that when transplant is about to take place, when the clone is used, there is a complete match of the tissues or organs that are used. You can just choose any of them. Really, you cannot say you forget the 3 or many others that we have learned. Let us now look at one scientific investigation question. This question, let's read together. I want to remind you, learners, do not run to answer the questions before you engage with the STEM, the notes. It's a table or a diagram that is given prior the questions, because the examiners will never put anything just to add up space on your question paper. Whatever they put has something to do with helping you to answer the questions correctly. Sometimes learners are too rushed, especially those that are high flyers. And here and there, they lose those marks that can improve their quality results even more. Let us read together. Male long-tailed widowbirds have extremely long tail feathers that they use in mating, displays to attract female. Scientists conducted an investigation to determine the relationship between the length of the male long-tailed widowbird's tail and its mating success. Because now the feathers, they say the longer the tail, they use that to attract the females for mating reasons. The procedure was as follows. Check the procedure. A total of 27 male long-tailed widowbirds were sampled and divided into 3 equal groups. 3 equal groups. The tail feathers of the tail in each group were treated in the following way. Now we've got 3 groups, and the 3 groups are equal. Interesting, isn't it? Then we've got group 1. Group 1, what happened is that the tail was cut short. And the second group made longer by adding artificial feathers. So instead of the natural long ones, there were artificial feathers that were added. And group 3, it was left unchanged. Group 3, the tail feathers were as they were, natural. Nothing had happened. Group 1, it was cut short, and group 2, there was an extension with artificial feathers, and group 3, that remained unchanged. Now, the 3 groups of the male long-tailed widowbirds, along with the female long-tailed widowbirds, were released into the environment suitable for mating. Each time a pair mated successfully, they produced a nest, and all the nests were counted. So you would see the mating pattern of the males and females, whether the mating pattern is more on the ones where the tails were cut short, or group 2 where they were made longer by adding artificial feathers, or group 3 where they were left unchanged. You read this not once, you read and read again until these things start to sink in your head and make sense. If you start understanding the investigation, it will be easy for you to engage with the questions. The average number of nests produced by each group was calculated and used as an indication of mating success. So the results are shown in the table below. Group 1, the results are 0.5. Remember group 1, that's where the tail was cut short. And group 2, I think if you check, group 2 has the highest. Group 2 was the tail that was even given an extension by the artificial colorful feathers. Group 2, the average number was 2,5. And group 3, which was unchanged, had the outcome of 1 nest in average. Question 2.1. Name the reproductive isolation that occurs in the long-tailed widowbirds. What is our answer? This is species-specific courtship behavior. Organisms can mate only if they are and produce fertile offsprings. Just remember what we learned in evolution about what a species is when they can produce fertile offspring. So this reproductive isolation is species-specific courtship behavior. Remember, there are 4 different types that we have learned, but on this one, we are just looking at species-specific courtship behavior. And then give us the independent variable of this investigation. Our independent variable here is, it's now the length of the male long-tailed widowbird's tail. That is our independent variable. This is the variable that can be manipulated. That is why you see from the 3 groups, what was manipulated? The length of the tails. One was cut short, the other one was extended with artificial feathers, and the other one was left unchanged. So this is the one that can be manipulated in order to get the outcome of the investigation. Explain why 27 long-tailed widowbirds were used instead of 3. Already, you know, in scientific investigations, basic things that we look at, we have got reliability, learners, we have got validity. But instead of 3, why did we have 27? And why? Because this increases the sample size. This increases the sample size and increases the reliability of the investigation. The larger the sample size, the more reliable the investigation. Don't forget that. Or the lesser or the lower the number, the less reliable the investigation. That is why generically, if you speak about reliability, it is to increase the sample size. And I want you to be extra careful. Learners have got maybe a tendency of just answering to say, oh, I know, reliability, increase the sample size and repeat the investigation. And as much as examiners play around how they ask the question, they give those 2 answers and they lose marks. For example, on this context here, you see it's the larger sample size. If you say increasing the sample size, yes, but why do we have 27 instead of 3? Then it is the larger sample size that was given. And then why do we have that? To increase the reliability of the investigation. Then explain why group 3 was included in this investigation. We had 3 groups. Group 1, the tails were cut short. Group 2, they were extended with artificial feathers. Group 3 remained unchanged. Why did we have group 3? And you know that group 3 unchanged, it's there to give us something that everybody should be knowing by now. And group 3, it is there because it serves as a control. So we see what will happen to the ones that were cut short in terms of the feathers, or to the ones that were extended. But this one remains unchanged. And let's see the outcome. It serves as a control so that it can be compared. So that it can be compared with the other groups. How many groups did we have in this context? We had 3 groups. So we're comparing group 3 with group 1 and group 2. And shows that the tail length is the only factor that affects the results, improve the validity of the investigation. So in other words, we are saying that the reason why we had the control group, which is group 3, it is to show that it is the tail length that is the only factor that affects the results. Because if we did not have the control, how will we know? Let's say we had two, one that was cut short and the one that was extended with feathers. The outcome that we had, will it be valid? No. So in order to look for validity, we must have control so that we're able to see that out of the mating successes based on the number of nests, it is only the length of the tail that had given more success or more failure in terms of what was expected in the investigation. I hope this is clear. Get this right, and this you cannot memorize. Memorize the basics, but then you must answer in context of what the examiner is asking you. There was a question that says, draw a bar graph to represent the results in this investigation. Learners, by now you know that when you are expected to draw any form of a graph, firstly, you write the caption of your graph. So let's check the caption of our graph here. The caption is the average number of nests produced by each group of birds, right? So this is our caption. You can see now coming to the criteria set for marking the graph. You see caption, both variables are included. So we see we've got the average number of nests produced, and we also have the group of birds. We have got the caption which is correct. You can also, you know, there are different ways. Some learners might choose to say produced by each group A, group B, and group C. That is still fine. It will give us a full story of your caption. Remember, in different scenarios, they might also have the units. Remember to include them on your caption, but here we are fine. And then our bar graph, there we have got group 1, we have group 2, we have group 3. And our y-axis is showing us the average number of nests produced. Here on the table did not have any units whatsoever. If there were units, you must include the units here. If you don't include the units, you lose marks for what? For your labeling. So an axis labels, X and Y axis correctly labeled. So it's a group here that is correctly labeled, and we've got the group that is correctly labeled here, and therefore you get a mark for your label. I want you to check very carefully. The scale for X and Y axis. Equal space between bars and width of bars for X axis and correct scale for Y axis. Learners lose marks because they concentrate sometimes on the x-axis and ignore the scaling on the y-axis. Let's engage with our graph that we have here. Check the width. Take your ruler. Do not do freehand drawing. You will lose these marks. These marks are free because they are skills-based. You don't have to be a rocket star to get the 6 marks for a graph. And the 6 marks will boost your marks big time. So you check the spaces. Okay, check the spaces here between 0.5 and 2.5. The space is equal with this space here. And when we speak about the width here of this bar is exactly the same as the width of this bar, and just the same here, right? Also, if you want to be a smart learner, just write the information that is given on the table. Like you can see group 1, the outcome was 0.5 and 2.5, and then for group 3, it was 1. This makes it easy for the person that is marking your graph to track down the bars and what they represent. But check the y-axis. The y-axis as well, the scaling is exactly the same. From 0 to 0.5 is the same, and just like that, as we carry on, you can see the spaces and the intervals are the same in order to get your 1 mark for scaling. Then plotting of bars, 1 to 2 bars plotted correctly, you get 1 mark. All 3 bars are plotted correctly, you have your 2 marks. There is no reason for you to go wrong. There's no reason for you to get bar number 1 and bar number 2 correct and you get bar number 3 incorrect. Take your time. You have 2.5 hours to answer a paper that is out of 150 marks. Do not cut corners. Get your marks. So this is how the graph is marked. You can see the plotting here is correct, the plotting there is correct, the plotting there is correct. We also, when we mark, we check, we come and say, okay, 1, it was 0.5, let's see, 0.5, okay, so the bar is standing by 0.5. Be careful, don't use freehand. Then we come to 2.5 and we check 2.5, we use a ruler. Here it's a freehand, don't copy it, madam. Right, and then we check the last one. And that's how you get your 2 marks. The same applies to all. Histogram, line graph, and a pie chart is different. You also must make sure that you bring all the relevant stationery when you write your exams. 2.5 states the conclusion of this investigation. Already you have seen the outcome. How do you draw the conclusion? The conclusion is drawn from what you saw. The table that we looked at gave us an indication of what we can conclude from this investigation. And what is the conclusion, class? The longer the male long-wigged tail, widowbird's tail, the higher the mating success. Yes, the higher the mating success. 2 marks. It's either you get 2 marks or nothing. Then, the shorter the male long-tailed widowbirds, the lower the tail, right? So you've got our tail there, the lower the mating success. 2 marks or nothing. Conclusion, you conclude from what you see. Method, you tell us how the experiment is done. And the aim is to see what was the aim of this farmer. The farmer's aim was to establish that the long-tailed widowbirds males, when they have got their long tails, then the success of the mating is higher. That's what you start looking at all those different things, and in that way you have answered your scientific question. Remember, 20% is there. Paper 1 and paper 2, so scientific investigation, dark or blue, high or low, is there. Practice, practice, practice. We are now going to have our quick stretching break, and let's meet soon. Just, I hope you got time to stretch and brace yourself. I could imagine some of you arguing about some of the things that we have discussed. Feel free to ask questions so that you can have clarity of whatever is confusing you. We were just starting with fossil evidence. So let's read together. Fossil evidence for humans may be interpreted in different ways. One possible model of human evolution is shown below. There are different ways. You might have seen them in different past papers, but look at the one that is presented. As per indication earlier on, once you see a diagram, do not take it for granted. Engage with it so that you understand what the diagram is talking about before you engage with the questions. So we have got here a million years ago, we've got 4, 3, 2, 1 present time. Then you see the types of how humans evolved, because this is now present time Homo sapiens. But you can see that from here, from the Australopithecus appearances, there had been a lot of changes over time until we moved and until we reached the present time. Engage with this. Look at this because it's going to help you to answer the questions. And the first question here is, name the family. Name the family to which all of the represented organisms belong. So we are looking at the Australopithecus and we are also looking at the Homo. Under which family? You must know. You must know the genus, you must know the groups, you must know the order, you must know the family. But under which family? The ones that we have here, do they belong? They belong under the family of Hominidae. Hominidae. Get the spelling right, because yes, spelling counts. Question 2, describe how cultural evidence is used to support the theory of human evolution, that humans evolved over time. What is it that we see from cultural evidence that is proving that there had been changes, these gradual changes that happen over time? Evidence such as tools, the use of tools, because humans now are able to use tools and weapons, and the language, we are talking, you are able to understand my language, and the artifacts that are used by humans, it's used to show advances in human development. We are communicating, you are able to hear me, we are really advanced. I can use the gadgets because these are the tools that I'm using to engage with you in this class. How long ago did the most common ancestor of Homo erectus and Homo heidelbergensis exist on Earth? How long ago? You need to check the common ancestry. So you come to the table here and you are going to look at this tree. So we are tracing Homo erectus. Okay, here is Homo erectus. And we are also tracing the Homo heidelbergensis. Right. Then the question is, how long ago? Right, did they exist? So the common ancestor, and let us see the answer there, it's 3 million years ago. The question is, do you understand how we get the 3 million years ago? Check the Homo erectus. You see where it started, and then the Homo heidelbergensis. If you see, it starts from the common ancestry was here, and now you can see it is showing 3 million years ago. This is easy. Sometimes the question can direct you to half of 3 million or half of 4 million. You must just take your ruler and establish the number so that you are correct. But the common ancestry on this particular one starts here in million years ago. And the next question is, explain the possible reason why the Homo ergaster was placed, between Australopithecus afarensis and Homo heidelbergensis. Why? So you come back here and say, okay, we are looking at Homo ergastus. Why was it placed between Australopithecus afarensis? So you look at, where is my Australopithecus afarensis? Here is my Australopithecus afarensis, and this model here of Homo heidelbergensis. Why was it placed between these two on the model? Why is this one between Australopithecus afarensis and, you see it's between the two, Australopithecus afarensis and Homo heidelbergensis. Why is Homo ergaster placed there? Because Homo ergaster shows the characteristics of both, right? A afarensis, that is Australopithecus afarensis, and Homo heidelbergensis. Therefore, it is a transitional species. Something that is found in between, it carries some. So because the development was starting from the most ancient 4 million years ago, where we had, not 4 million years ago, 3 million years ago, when we had Australopithecus afarensis. But gradually as you move, you can see that Homo heidelbergensis, it's after 1 million years ago, maybe 500 million years ago, not 500,000, not 1 million, right? And then if you see Homo ergastus, this is now somewhere here. You can see it's between 1 million and 2 million, but then Australopithecus afarensis was the most ancient in 3 million, and then this Homo heidelbergensis is 500,000 years. So you can see that between this one, it's a transitional, we call it a transitional fossil, carrying the characteristics of the most ancient and the most recent. Explain how the fossils of organisms that existed from 4 million years to the present time are used to support the out of Africa hypothesis. Out of Africa hypothesis. Be extra cautious when you answer this. So we are talking about the fossils of organisms that existed. So those that have existed long time ago to the present day, the present time, they are supporting out of Africa hypothesis. And let's have a look at the answer. Our answer there is that the fossil of Australopithecus were only found in Africa. You cannot say they are found in Africa. Please remember to emphasize that the fossils, the remains of the Australopithecus, they were only found in Africa. You have a mark. And you can also add that the fossil of Homo habilis were only found in Africa. So we're talking about the Australopithecus, and we are now talking about Homo habilis. It's also only found in Africa. And the oldest only found in Africa. And the oldest fossil of Homo erectus were found in Africa. If you just say that the fossils of Homo erectus were found in Africa, you are losing that mark. You are losing the essence of the answer, because we are talking about the oldest. Remember, Homo habilis existed before Homo erectus. So the oldest fossils of Homo erectus were also found in Africa. And the oldest fossils of Homo sapiens were found in Africa. And this suggests that the ancestors of Homo sapiens originated in Africa. So you see that we, following the lineage, we are talking about the Australopithecus, right? We have got Australopithecus africanus, Australopithecus afarensis. However, we are just saying the fossils of Australopithecus were only found in Africa. And then we speak about Homo habilis, that they were also only found in Africa. Then we speak about Homo erectus, they were only found in Africa. But the Homo erectus, the oldest, and also Homo sapiens. Because if we say the fossils, we are Homo sapiens, the remains of the most recent ones, they are found all over, not only in Africa, but the oldest. So Homo erectus and Homo sapiens, remember to say the oldest, were only found in Africa. And this suggests that the ancestors of Homo sapiens originated in Africa, and this supports out of Africa hypothesis. Please just get these free marks. It's straightforward, it's easy. Just get the 3-4 lines right, then you get your marks. Now let's look at this other question. Potos and lemurs are small mammals. Scientists believe that potos and lemurs share a common ancestor that existed in Africa. So we are looking at common ancestry that existed in Africa. Presently, potos only occur in Africa and lemurs are only found in Madagascar. Madagascar is an island of the east coast of Africa, as shown in the diagram below. So you look at the diagram, you can see that Madagascar, yes, Madagascar. So Madagascar is just an island which is off the east coast of Africa. So this is the eastern coast of Africa, and we have Madagascar. Question 2.1. Explain how continental drift could have affected the distribution of common ancestry. Just for you to remember, we have learned that the Earth was one big mass, which was called what? Pangaea, which now split into two big masses, which were Laurasia and Gondwana land. Then we had more splits that resulted in the continents. Now you are explaining. I want you to always remember to check the doing word, the verb of the question, so that you are able to answer in context of what the examiner is asking you. So there was once a large continent, yes, we know about Pangaea, and the common ancestor existed throughout that continent. So the common ancestor were existing in Pangaea. Right, and when Madagascar separated, you see Madagascar is now separated due to continental drift. The common ancestor was found in both regions. So whatever existed in one piece. Now when the splitting took place, some of the common ancestors were found here and some were left on the east coast of Africa. That is our explanation. Describe the speciation of potos and lemurs to become different species. How many marks do we have here? 6 marks. Speciation, generally you will find 6 marks or 7. Darwin's theory of natural selection, it's 6 marks, and you have got very good marks also on Lamarck's theory. But let's look at speciation, the formation of new species. But remember, there's a generic one that you have been taught, but you answer in context of what you are being asked on the question itself. So let's have a look. The common ancestor became separated into 2 groups by the ocean. You can see that we've got Indian Ocean, which has separated the common ancestor, because we earlier said common ancestor is found here on Madagascar, and part of here has got the common ancestor on the east coast of Africa. And then there was no gene flow between the 2 groups. So what was found in Madagascar could not have any gene flow with what was found on the east coast of Africa. There was no gene flow between the 2 groups. Each group experienced different environmental conditions. The group that is here is an island. The environment is different. So both the one that is on east coast of Africa experienced different environmental conditions compared to the one that was in Madagascar, the group. And they underwent natural selection independently. When you speak about this, don't forget to mention the independence, because natural selection for those ancestral common ancestors that were found in Madagascar, they underwent their own natural selection independent of what was happening on the common ancestor that was found on the east coast of Africa. Then you say they underwent natural selection independently. The individuals in each group became different. Now, after the independence that has happened, now the 2 individuals, the ones in Madagascar and the ones in the east coast of Africa, they became different. Genotypically and phenotypically. Do not just write phenotypically only. So they changed completely with their genes and also how they looked from outside. Sometimes examiners will choose to give you a mark for genotypically and also phenotypically, but this one is choosing to give you 1 mark. So if you had something missing, then you are going to miss that mark. To form potos and lemurs. Eventually, if the two groups are mixed again, so let's say, for argument's sake, the group that was found here was put here on the east coast of Africa to mix, what will happen? They can never interbreed, or they will not be able to produce a fertile offspring, because remember, the same species must be similar and it must be able to produce fertile offspring. They must be able to interbreed and produce fertile offspring. So if there are now two different species that resulted, they will never be able to produce fertile offsprings or even to interbreed. You have your clean 6 marks. These marks you cannot afford to lose. These marks you walk in the examination room holding them in your right hand. We have a table below shows information on selected hominid fossils. What do? You check the table as per indication. So you're checking the table here. So we've got common name of fossils, and we've got species, and we have fossil site, scientists responsible for discovery. I just hope you have learned about all the different scientists that have been involved in discovery of different fossil remains. So common name here, we've got P, but we've got Dawnchild, and species Australopithecus sediba, and where Malapa cave in the cradle of humankind, and the scientists responsible, we don't have a name for Q, and Dawnchild, we don't have the species name, but it was found at Stekfontein caves, and we don't have the scientist. So question 1, name fossil P. We are looking at fossil P. What is fossil P? Which is the species of the Australopithecus sediba? It's Garabo. Garabo, Australopithecus sediba. Then identify the species at R. So you come here, okay, it's Dawnchild. Dawnchild, by the way, which species does Dawnchild belong? Right. And our answer is Australopithecus africanus. This is the information that you must memorize. You must know the common names of the fossils, you must know their species names, you must know the sites where they were discovered and the scientists that discovered them. It's straightforward memorization, this one. Nothing will change the answers. So it's Australopithecus africanus. You can see that we have got a capital letter as we start, and we have got a small letter there. Be careful and underline when you do your species names. Give the name of the scientist at Q, and if we look at Q, who discovered Garabo? Everybody knows it's Lieberger. And now, who discovered, we're looking at S now, who discovered the Dawnchild? Who discovered the Dawnchild is Raymond Dutt. Let us now look at something different. The table below shows some dog breeds and their desirable characteristics. You look at the diagram, you look at the table, and you read. We are reading together. Dogs have been selected and bred over many years to produce approximately 340 different dog breeds. They have been bred for certain characteristics desirable to human. An analysis of 736 base pairs of the cytochrome B gene showed that gray wolves are the only direct ancestor to the present-day dog breeds. All dogs breeds belong to the species Canis familiaris. You have read, don't read it once. I'm reading it once, but read it twice and read it the third time so that it starts to make sense or to speak to you. But again, come to the table and see what you are given. Okay, so what is this now? Okay, I see. This is the gray wolf and this is the ancestor. Okay, but now due to human intervention, there have been desired characteristics that they wanted. So we've got a little poodle, which is very intelligent. We've got this pug here, loving, affectionate, and playful. And we've got Saint Bernard, outgoing and adventurous. Rhodesian Ridgeback. This one is strong, is athletic and fast. Excuse me. Then, name the process whereby the different breeds of dogs were produced. So, even without you reasoning, you have been told earlier on that it was human manipulation that resulted in the breeds that we have today from the grey wolf. But now you tell us the name of the process. And what is that process? The process is called artificial selection or selective breeding. Yes, it's artificially selecting the specific desired genes in order to have the desired outcome. This is done by men. Describe how humans carried out the process named in question 2.1. You are now going to describe how humans carried out this process of artificial selection. So let's see how they carried this process. They chose dogs with desirable traits and interbred them. So they firstly, you must know what you want. You cannot be a scientist that just go and take anything and you say, I'm trying to get something that you don't even know. So they chose dogs with their whatever desirable traits that they wanted. And what did they do? They interbred them. Remember, this process is not like in one generation. It takes time. It's over several generations that we can get the desired outcome. I'm just mentioning this for your information. And to produce the offsprings with this trait. So over generations, the offsprings with the desirable trait were produced. Explain how it can be confirmed that all these dogs breeds belong to the same species. So you are looking at different breeds, right? But now, are they belonging to the same species? Can you see that the examiner can just play games with your mind sometimes? Because they are saying, you explain how it can be confirmed that they belong to the same species. But already, you have learned about the definition of a species. Species is similar organisms that are able to do what? To interbreed and produce fertile offsprings. So let us see what the examiner was looking for. The examiner was looking for allowing them to interbreed with each other and see whether they produce fertile offspring. So in other words, this is just a different approach to you knowing the definition of a species, because if it is a species, it means that a species must be able to do what? A species must be able to interbreed. That's the first thing. And a species must be able to produce a fertile offspring. So if ever breeding has taken place and the species does not produce the fertile offspring, then we have a problem, because sometimes it is possible that like a donkey and a horse, the outcome that we got was a mule, but a mule is infertile. Therefore, it means that the donkey and the horse are not one species. Because once there must be any form of interbreeding, the offspring must be fertile. So in other words, this offspring, the outcome, the offspring that is being produced must be able to carry on and produce more offsprings because it is fertile. If there's infertility, then we see that that is not a species. So in other words, what is meant, because they said, explain how it can be confirmed that all these dogs, you are looking at all these dogs that are mentioned here, right? How it can be confirmed, it's because this dog can mate with this, and this one can mate with that, and this one can mate with that. Or an analysis of DNA is made to check for matching sequences. You know that the DNA will always give us an indication that the sequence that is given is matching, it's correct. And if the sequence is not matching, then you can see that this is not compatible. They cannot interbreed. They cannot produce a fertile offspring. So either you speak about the interbreeding part and the producing of the fertile offspring, or you can speak about the analysis of DNA and see if the sequences are matching. Explain which of the dog breeds shown would best be used for hunting. Out of 1, 2, 3, 4. Which one? How will you get the answer? You are looking at the characteristics. Intelligent. This one cannot hunt, but smart. Pug. This one is just loving, affectionate, and playful. But if you see, now check the Rhodesian Ridgeback. This one is strong. It's athletic and fast. Therefore, as you explain what you tell us that the name of the type is a Rhodesian Ridgeback. Then you tell us what makes you, what justifies, because you must explain to us. The Rhodesian Ridgeback is strong, athletic, and fast. Why do you choose to leave one of the characteristics that are made? Mention all of them to get the mark, because you will never know if the examiner is going to be fussy that it was given for the learners. They should have mentioned all to get that one mark. And it's able to catch the prey, because it's now for hunting check. So you can safely say that it will be able to hunt and catch its prey. Explain how the present day dog breeds may be disadvantaged in relation to their common ancestor. What is it that might be a disadvantage for what we have today compared to their common ancestor? Who is the common ancestor, by the way? Is the grey wolf. Because now, when they are the present day ones, they have got the reduced gene pool, narrowed into what was desired. And this also had allowed these variations. They are now varied, right? Due to the reduction of gene pool or variation, they will not be able to hunt or find shelter or defend themselves as well as wolves are able to. As much as you see all of them, they originate from this common ancestor. But you can see that some of them, the intelligent one, little poodle will not be able to maybe defend itself like the wolves, because the wolves can still do all this. Therefore, unable to survive in the wild. If you take little poodle to the wild, intelligent as she is, might not be able to survive in the wild. Let us look at this diagram here. I am not going to tell you what the diagram is. The diagram below represents the evolution of the family Hominidae. We spoke about the family earlier on, where we had different organisms that were outlined. And again, what is the rule? The rule is that you study what you are given. So you come to this. What is this? Okay, let's see. All right, so I have got P, I have Q, there is R, I have S. Okay, then you check this line. Just make sense. Try to make sense. This long line here that connects here is orang-utang. And then we have got gorilla, which is here by R. And then there's bonobo. We've got humans here. Okay, there's humans there. And we've got bonobo and chimpanzee. Just try to figure out what is it that might come my way as a question. Very important. Question 1. Name the type of diagram represented above. Everyone knows this answer. You must know this answer by now. What is this diagram called? It is called the phylogenetic tree or the cladogram. Check the spelling. Learners have got sometimes a weakness of failing to say it fully. They'll write phylogenetic tree or phylogenetic tree and not phylogenetic tree. You get your full mark by telling us it's a phylogenetic tree. And because the question said the diagram, that is why the tree is part of your answer and you have your tick there. Then question 3.2, give the letter of the organism which is the common ancestor of all hominids. What are you giving us the letter? So the common ancestor of all hominids that are mentioned here, which letter shows us the common ancestor? P. How do we see? You can see that this is now, according to the phylogenetic tree, P is the most primitive. And all the other branches, they emanated from P. So our common ancestor is P. Now give the letter of the organism which shares the most recent common ancestor with the gorilla. So you are going to check your gorilla and you must give us the one that shares the most recent common ancestor with the gorilla. And what is that? Our answer is R. You can see, right, gorilla is there and R is showing that they are sharing the most common ancestor. You can see the line that is connecting the gorilla and R, and our answer is R. Don't confuse yourself and start writing names. We were asked to give the letter, just as a reminder or a disclaimer. Again, on the phylogenetic tree or cladogram, name two organisms that A have S as a common ancestor. So you come to this cladogram or phylogenetic tree, name two organisms that have S as a common ancestor. Here is S here, and our S is the ancestry. But then how many organisms are we looking at? You are naming two organisms that they have this S as their common ancestor. And then let's see what your answer is going to be. It is bonolo and chimpanzee. But we don't guess. So we are going to check our phylogenetic tree. Here is S, and the S has this line here. But if you check, the line gives rise to two organisms. And the two organisms that we have bonolo and we have chimpanzee. So for you to name two organisms, because the question was asking for two organisms, and your answer is going to be bonolo and chimpanzee. You can check anything. You see, they can even sometimes from the phylogenetic tree, they can ask you between the orang-utan and bonolo or gorilla, which one is the most primitive or the most recent. So what you do, you check your connecting lines on your phylogenetic tree. You see this one is directly connected to the most common ancestor. And if you check gorilla, you see gorilla, basically gorilla had emanated after orang-utan. That is why you can see that it has got this line that gives us the break up to gorilla. And then now gorilla break up with R, and then we carry on, there's another breakup of S, and then we have this. All these lines, they speak, they connect. They have got a way of connecting to tell you or to show you that there is a relationship. Hence we speak about a tree, phylogenetic tree or cladogram, showing us the evolutionary relationships and how long ago organisms have existed. The diagram below, it's still the same one of the family Hominidae. Question 3.3, name two organisms that are quadrupedal. Quadrupedal are the organisms. We've got bipedal organisms and quadrupedal organisms. Quadrupedal, they are using both four limbs to walk. But how many organisms are we looking at here? You mentioned only two organisms that are quadrupedal. So which organisms are quadrupedal from this? We have got the orang-utan, it's quadrupedal. We have gorilla, quadrupedal, and we have bonolo, that is quadrupedal. We have got chimpanzee, that is quadrupedal. And the question is, if you look at this phylogenetic tree, which one is the present time one? So you can see that these lived primitively, bonobo and chimpanzee, hence they were still quadrupedal. But if you look at humans, check the line, yeah, the humans are the most recent organisms. And humans are not quadrupedal, but they are bipedal. And just as a reminder, what is the advantage of bipedalism is that the organisms they have got a bigger surface area, and also to say that they are able to view far, to see danger from far, and their hands are free to carry objects or to carry babies. That is the advantage of bipedalism. And you know that there are characteristics that separate the quadrupedal organisms and bipedal organisms. Hence, we are looking at this question now, which says, describe 3 anatomical features of the skeleton of the quadrupedal hominid. It's a description. 3 anatomical features. Don't get confused. This information you know. Quadrupedal hominids, what makes them to be quadrupedal? It is because the foramen magnum is at a more backwards position. That opening on the skull is more at the backwards position, meaning that they are forced, quadrupedals are forced to be like this, because the foramen magnum is connecting from a more backwards position. And they've got a C-shaped spine. Because they walk like this, they have got a C-shaped spine. They cannot have an S-shaped spine like the bipedal organisms. And they also, just in close, they also have got a narrow, and a longer pelvic girdle. So that allows them to be quadrupedal. And on that note, we have come to the end of our very interesting revision. Keep on keeping on. Till we meet again. Bye-bye.